Hello, my name is Natália Koch, and I'm a postdoc researcher at the University of Minnesota, United States. Today I'm going to present my work entitled Nuanced Qualitative Trait Approaches Review Strong Environmental Filtering and Phylogenetic Constraints on Lichen Communities. This work is co-authored by Daniel Stanton, Sandra Miller, Adriano Spielman, and Robert Lucking. So first I'm going to talk a little bit about the differences between quantitative and qualitative traits. Quantitative traits are ideally directly related to the function of interest, for example, water holding capacity and moisture availability, while qualitative traits are usually surrogates of a certain function, for example, the type of photobiont with light, humidity, and temperature conditions. Functional trait approaches are very powerful when there is a well-established framework of trade-offs. What is the case, for example, of plants and some groups of animals. However, universally measurable quantitative traits are a challenge for lichens because of the greater variation that they have of different growth forms, for example. And that's why most studies are on macrolichens using quantitative traits. So I think that a nuanced qualitative trait approach has the potential to reveal overlooked patterns with normal qualitative trait approach, especially in a phylogenetic framework and including all types of lichens. So with this work, we aim to understand how environmental filtering and phylogenetic constraints act on lichen community assembly along natural forest succession, and to evaluate the use of a functional approach based on nuanced traits. So in 2013, we published a paper on functional diversity and traits assembly patterns of lichens as indicators of successional stages in tropical rainforest. And we found that fruticose lichens and foliose lichens, as well as lichens with lirelli, were more related to initial stages of forest succession with higher canopy openness, while lichens with pyrithecia and squamulose lichens were more related to late successional stages with low light availability. We used the same database that were based on lichens sampled on tree trunks in 3 different successional stages from 6 to 60 years of forest natural recovery. To sample the lichens, we used the rubber band method that was described by Marcelli in 1992 and sampled a species cover from 30 to 150 centimeters from the ground. We established our sampling units in 4 valleys in the city of Maquiné, South Brazil, state of Rio Grande do Sul, here in the map. And they are these areas are composed by Atlantic Rainforest, and we had 88 sampling units per stage and 3 trees sampled at each sampling unit. In total, we had 24 sampling units and 72 trees sampled. So the areas, the forest successional stages varied when we looked at tree density, trunk basal area, and canopy openness. And you can see these differences when looking at these pictures. Here is the initial stages, how they look like inside the forest, the intermediate stages, and the late stages. So that's an example of how they looked. So to do this work, we used several lichen functional traits, and we categorized them in as much as possible attributes that we could do. So for example, we had the type of photobiont that we categorized in green crocoid, green tedepolia, and cyano. Photobiont genus that we categorized based on the genus in the genera that we found, growth form that we broke into crustals attached, loosely attached, narrow lobes, foliose, wide lobes, microfoliose pressed, gelatinose, filamentous, fruticose, and squamulose. We also had the upper cortex type, the main type of reproduction, the main reproduction strategy, the type of ascomata disc, the size of propagules, the form of ascospores, the cortex substances, medullary substances, terpenes that are an important class of medullary substances, ascomata chemistry, and farina. So we also built a phylogenetic tree for the 122 lichen species that we found based on recently published phylogenies. And here we color-coded these trees based on, we color-coded the clades based on the orders that they were related to. As results, we found and we detected changes in taxonomic, functional, and phylogenetic composition, as well as in structure and diversity along this gradient. So here in this figure, I just show some examples. I just highlight some examples that we found differences among orders and within orders. So Ostropales, for example, had different families that were found in initial and intermediate and late stages of forest succession, with, for example, Graphidaceae with higher abundance in initial stages, while Polynesia had higher abundance in late stages. In Calicialis, Physiaceae was only found in initial and intermediate stages, and another example in the Leucanuralis, Hamalineaceae was found in higher abundance, in initial, in intermediate and late stages, while Parmeliaceae was only found in initial stages. So when you look at the NMDS with lichen species composition, we can see that initial, intermediate, and late stages were completely different based on their cover and species composition. So initial stages, as I mentioned before, had higher canopy openness, while late stages with older forests had also a greater trunk basal area and lower canopy openness. So here in this box plots, I summarize the results, most of our results, and what we can first see and first mention, it's important to mention that the sites varied, as I said, regarding their age, the different forest succession stages, the sum of trunk basal area and the canopy openness. So higher trunk basal area and lower canopy openness in the late stages, which reflected in differences in species composition, as I showed in the NMBS. And here I'm just plotting the first axis of this NMBS just to summarize the result. We also found differences in functional diversity and phylogenetic diversity, where late stages had lower functional diversity, while initial and intermediate stages had higher functional diversity, and the same pattern was seen in the phylogenetic diversity. We found no differences between the net related index and nearest taxon index, which is related to how phylogenetically grouped these communities are. So they didn't vary, the stages didn't vary, but all the groups, we can say that they were phylogenetically clustered. We also saw that species diversity was greater in initial and intermediate stages, and species beta diversity has the same pattern when we look at within patterns, within the communities, within the sampling units. But when we compare between units, between stages, we can see that intermediate and later stages, they had lower difference between them. So the areas were more similar when we look at species diversity. By looking at the traits, the functional traits that we found differences when looking at all the traits that were evaluated. So we found differences among the stages, and when we look at the photobiont genus, when we look at the growth form, when we look at the upper cortex type, which is something new, the propagule size, the cortex substances, the medullary substances, and the pruina. So all the traits that we evaluated changed from initial to late stages of forest succession. We can highlight, for example, that green algae had different patterns when we compare the genera, which is very interesting, and also the growth forms. Looking at the nuanced traits, we can see different patterns, for example, for gelatinous lichens, foliose wide and foliose narrow among these successional traits, successional gradient. And important to highlight as well is that cortex substances and medullary substances and purina were only and mostly seen at initial and intermediate stages and not found at late stages of far succession. So we also made partial mantle tests excluding the effect of phylogeny to see if the patterns were still the same. So we found that functional traits such as photobiont genus, type of cortex, reproductive strategy, propagule size, and protection strategy showed strong responses to succession even when controlled for phylogenetic relationships, which is shown here in this table with the statistics and the p-value. So as take-home message, we can highlight that mature forests with closed canopy structure, they work as a strong environmental filter for understory lichens. And that's important. We are saying here that this filter is for understory lichen, and we are not evaluating the canopy lichens that probably surely are there, and the diversity is completely different. We imagine that. So this reflects in species turnover in the understory, limiting phylogenetic, functional, and species diversity, and also reflects in unique functional and phylogenetic composition. So the use of a nuanced qualitative trait approach may overcome some limitations of categorical traits and can reveal overlooked key functional attributes, as we showed, for example, related to maybe carbon concentrating mechanisms for the types of algae photobionts, cortex anatomy and chemistry, and propagule size. So I would like to thank my co-authors, Dr. Robert Lucky, Dr. Sandra Miller, Dr. Daniel Stanton, and Dr. Adriano Spielman for all their support and help along the process. My co-advisor for the master and the PhD, that is Susana Martins, showed here, and my friend and my companion and my assistant in the field, Fabiana Luqueta, and all the institutions that were involved with this work. Thank you so much, and I'm open for questions.